Genetic metabolic brain disorders, GMBDs, are neurological conditions caused by inborn errors of metabolism, IEMs. These disorders typically occur due to gene mutations, leading to a deficiency or dysfunction of metabolic enzymes. This disrupts normal metabolic processes. When metabolic products accumulate in the body, especially in the brain, it leads to neurodegenerative changes, cognitive decline, and motor disorders. Common IEMs include lysosomal storage diseases, mitochondrial diseases, lipid metabolism disorders, and other related conditions. The mechanism behind GMBDs involves the accumulation of metabolic products in the brain and body. This causes neuronal cell death, neurotransmitter imbalances, and disrupted neural circuits, which severely affect cognition, emotions, and motor functions. It may also lead to mental health issues like depression, anxiety, and psychosis. While current treatments mainly focus on correcting metabolic abnormalities, emerging therapies such as stem cell therapy and gene therapy offer new hope for treating inborn errors of metabolism-related brain disorders as science and technology continue to advance. This video will explore the connection between GMBDs and nervous system damage. It will dive into how metabolic abnormalities can trigger cognitive, motor impairments, and migraines by affecting neurotransmitter balance, cerebral vascular function, and neuroinflammation. Additionally, we'll discuss the potential of stem cell therapy and integrated treatment methods in GMBDs. Neuropsychiatric symptoms of GMBDs. Neuropsychiatric symptoms in GMBDs can include cognitive impairments like learning difficulties, memory loss, and disorientation. These symptoms typically appear in childhood or adolescence and may progress to dementia. Psychiatric symptoms, including emotional flatness, mood instability, depression, anxiety, and hallucinations, are common and often appear with cognitive decline in adulthood. These symptoms are linked to metabolic dysfunction in specific regions of the brain. Common motor impairments in GMBDs. Motor impairments often include poor coordination, ataxia, lack of muscle control, tremors, and Parkinsonism. These symptoms typically result from damage to the cerebellum, basal ganglia, and corticospinal pathways. Around 50% of patients may experience seizures, which often don't respond well to anticonvulsant medications. Additionally, sleep disturbances and emotional instability are common, especially in the chronic phase, where patients experience significant mood fluctuations. Migraine in GMBDs. Migraine is a common neurovascular disorder in GMBDs, often characterized by severe headaches, nausea, vomiting, and sensitivity to light and sound. In GMBD patients, migraines are closely linked to the accumulation of metabolic products and nervous system dysfunction. Key mechanisms include neurotransmitter imbalances. Disorders like mitochondrial diseases and amino acid metabolism disorders can imbalance neurotransmitters like serotonin, leading to more frequent and intense migraines. Cerebrovascular dysfunction. The accumulation of certain metabolic products can damage the smooth muscle of cerebral blood vessels, interfering with blood flow regulation and triggering headaches, especially in lysosomal storage diseases and some mitochondrial disorders. Metabolic product accumulation. Products like lactate, ammonia, and cholesterol can accumulate in the brain, leading to cerebral edema and neuronal damage, both linked to migraines. Neuroinflammation. The buildup of metabolic products can activate inflammation in the brain, which is strongly associated with migraine onset, especially in patients with neurotransmitter imbalances. In mitochondrial diseases, like MELAS, migraines are a common symptom. Mitochondrial dysfunction affects the nervous system's energy supply, leading to neuroinflammation, oxidative stress, and abnormal cerebrovascular responses, all of which trigger migraines. Amino acid metabolic disorders, like phenylketonuria, can also cause migraines, primarily due to the accumulation of metabolic products and neuroinflammation. In lysosomal storage diseases, Migraines are often accompanied by other neurological symptoms such as epilepsy and dystonia, with lipid accumulation in the brain potentially increasing headache frequency. The trigeminal nerve and migraines. The trigeminal nerve, CNV, is the main sensory nerve of the face, and its activation plays a key role in migraine pain. Energy metabolism disorders, neurotransmitter imbalances, metabolic product accumulation, and neuroinflammation may lead to abnormal trigeminal nerve activation, which can exacerbate or trigger migraines. The mechanism of brain region damage in GMBDS is typically due to the accumulation of metabolic products from IEM diseases, such as cholesterol, lactic acid, and amino acids in the brain. These accumulations may trigger functional impairments in brain regions through various mechanisms. IEM diseases lead to the excessive accumulation of metabolic products, such as cholesterol, in the brain. These substances are toxic to neurons, causing neuronal apoptosis. Purkinje cells in the cerebellum, as well as neurons in the basal ganglia and cortex, are particularly susceptible, which in turn leads to motor coordination impairments, cognitive decline, and emotional instability. Manifestations of genetic metabolic brain disorders, GMBDS. IEM diseases can lead to abnormalities in the synthesis and metabolism of neurotransmitters, such as dopamine and glutamate, which affect the normal function of the brain's neural networks. This, in turn, leads to cognitive, emotional, and motor function impairments, manifestations of genetic metabolic brain disorders, GMBDS. Metabolic disorders in IEM diseases may cause demyelination, particularly in the cerebellum and cerebral cortex, leading to motor impairments and cognitive decline. Metabolic dysregulation may also trigger brain inflammation, further exacerbating neuronal damage. Brodmann area and GMBDS. Brodmann areas divide the brain's cortical regions into distinct functional zones responsible for cognition, emotion, and motor functions. Metabolic dysregulation in genetic metabolic brain disorders, GMBDs, may affect these key regions, leading to widespread neurological impairments. Prefrontal cortex, Brodmann areas 9, 10, 11. This area is responsible for high-level cognitive functions such as decision-making, planning, emotional regulation, and social behavior. Metabolic dysregulation in GMBDS may affect this region, leading to emotional instability, difficulty with impulse control, attention deficits, and executive function impairments. Symptoms typically present as mood fluctuations, impaired decision-making abilities, and a decline in organizational and planning skills. Hippocampus, Brodmann areas 27, 28, 35. The hippocampus is closely related to memory and learning, helping to convert short-term memory into long-term memory. The accumulation of metabolites damages hippocampal neurons, leading to impaired short-term memory and even loss of long-term memory. Patients may exhibit cognitive issues such as learning difficulties, disorientation, and memory loss. Basal ganglia, Brodmann areas 4, 6, 8, 9. The basal ganglia are involved in motor control and coordination, regulating fine motor movements. When GMBDS affect the basal ganglia, it leads to motor impairments such as tremors, dystonia, and ataxia, indicating basal ganglia dysfunction. Neuronal damage caused by metabolic accumulation impacts the neural circuits in this area, resulting in a decline in motor control abilities. Cerebellum, Brodmann areas 17, 18, 19. The cerebellum is responsible for coordinating body movements, balance control, and fine motor skills. Metabolic disorders affect cerebellar function, leading to motor incoordination, unsteady gait, and other ataxia symptoms. The loss of Purkinje cells is a hallmark of cerebellar dysfunction. Stem cell therapy for GMBDS. Stem cell therapy is still in the research phase for GMBDS and has not yet become a standard treatment. While stem cells show potential for neurorepair, their effectiveness and safety are still being studied. Challenges with stem cell therapy. IEM diseases cause metabolic product accumulation in the brain, leading to neurodegeneration. Stem cell therapy can repair damaged neural tissue, but unless the underlying metabolic abnormalities are corrected, the repairs may only be temporary and won't fundamentally improve the condition. Research on stem cell-derived exosomes has shown some promise in animal models for repairing brain damage, but these studies mostly focus on non-genetic brain injuries. Therefore, the results may not be directly applicable to IEM patients. Stem cell therapy faces challenges such as treatment timing, stem cell sources, administration routes, and immune rejection, with no clinical trial data supporting its use for IEMs. Currently, treatments should focus on correcting metabolic abnormalities, such as gene therapy and enzyme replacement therapy. Conclusion: genetic metabolic brain disorders, GMBDs, are caused by inborn errors of metabolism, IEMs, leading to the accumulation of metabolic products in the body. This accumulation affects brain function, causing cognitive, motor, and psychiatric symptoms. While current treatments primarily focus on correcting metabolic abnormalities, emerging therapies like stem cell therapy offer new hope for better outcomes. Early diagnosis and intervention are crucial for slowing disease progression and improving quality of life. As research advances, we hope that new therapies will provide better solutions for patients.